Hello, our group is researching the effects of heat shock and rapamycin treatments on transcription levels in yeast. Cerevisiae has a similar aging process as humans and therefore serves as a good model organism to test drugs speculated to reverse aging, such as rapamycin, a drug that affects protein levels that's currently being used to treat cancer. In yeast, gene regulation is altered when an organism is exposed to high levels of heat in order to prevent cell death. We speculate that there may be a link in the genes affected by both heat shock and rapamycin treatment, since both affect protein synthesis.
We used 3 gene expression datasets from RNA-sequenced wild-type S. cerevisiae cells, a control group, heat shock treatment group, and rapamycin treatment group. Using RStudio and GoTerm analysis, we ran correlation tests between the expression levels of the different treatment groups. We found that heat shock and rapamycin treatments upregulated and downregulated similar genes. For downregulated genes, there appeared to be a large overlap in gene groups, while for upregulation, there was noticeably less of an overlap. This graph led us to believe that genes further from the origin of the graph, which had greater changes in gene expression levels, play a larger role in regulating stress.
After establishing a direct relationship between the upregulated and downregulated genes in both treatment types, we ran a GoTerm enrichment analysis with the highest and lowest gene expression levels. Our study found genes involved in the glycolysis pathway to be downregulated by heat shock and upregulated by rapamycin. Another group of S. cerevisiae genes were independently downregulated by both rapamycin and heat shock treatments, and they were found to be human homologs of the Huntington's disease pathway.
By understanding the regulation of genes treated with heat shock and rapamycin independently, we were able to identify the molecular mechanism and associated pathways of gene groups that regulate stress response. Future researchers can directly humanize yeast with diseases involving the accumulation of misfolded proteins and explore rapamycin as a medical treatment. This could suggest that the glycolysis pathway is closely involved in the progression of Huntington's disease.